I've chosen to talk through a separate science paper purely because I want everyone to be able to revise from this video. For those of you who are doing combined, your paper will start with easier questions. And towards the end, I'm going to tell you when it's going to be just content that separate sciences will need to do. For those of you doing combined, you wouldn't get to the end of the paper. So again, if you're doing foundation, there would be some more easily accessible questions before this. However, you still would have this in your paper. So I'm going to go through it for everyone. It starts by saying this question is about photosynthesis. Complete the word equation for photosynthesis. In this one, people get confused all the time. I think they panic because it's an exam and they stop thinking clearly. And what I mean by clearly is if you just picture, we're talking about a plant here. So what do you have to do if you've got a plant at home? First thing that you have to do is water it. It can't grow without water. And hopefully you remember that out the other side, and they've given it here, we've got oxygen, and we know that we need the oxygen from the plants to live. Therefore, the gas that they've got going in has to be a different one to oxygen, which is carbon dioxide. I've put that as one word just to cram it in. Why does a plant go through photosynthesis? What is the benefit to the plant? It has to make glucose. So that is your equation. You also need to know the formula equation, which is 6 molecules of H2O. Carbon dioxide, there is 6 CO2. Glucose is C6H12O6. There's just one of that because it's so large. And again, 6 molecules of O2. Describe how energy for the photosynthesis reaction is gained by plants. So for this one, the kind of two key words we've got here is, in my mind, the fact that you said, what is the energy? And if you remember, it's talking about this bit here. What is the thing that powers the process? You guys in chemistry have probably talked about words like exothermic and endothermic. Photosynthesis is an endothermic reaction, and that's something that you do need to know, which means that it takes in energy. So the energy it's taking in, we know, is sunlight. So you have to have said sunlight to get this question. Oh, if I could spell it, that would be great. How is it gained? So sunlight can't just hit the leaf. It has to be, dragged into the plant almost somehow. And hopefully you guys will remember that's by the chlorophyll. So that is by the chlorophyll, and that is a pigment found inside chloroplasts. So I'm making sure that I put both things in just to ensure that I've got the mark, but chlorophyll is the answer that they wanted. That's how sunlight is absorbed. You're then given this table, as you can see here, and they have put a value here as an X. They haven't calculated the mean for you. So already I'm thinking the mean's got to be calculated. Hopefully you guys remember mean is you add up each of your values and then divide by how many there are, so it will be divided by 3. When I have a look at this, the first thing that I was thinking was, when I saw this paper, do they want you to ignore that value? So sometimes they'll throw in a value that's far too high, far too low, and it's almost like trying to trick you to understand if you do get the data. If there's something that doesn't fit the pattern, it shouldn't be included in the mean. However, when I look further down, you can see here they do have a massive gap of between 32 and 34, that's 2, and they've included that when they calculate the mean. So the fact that that's 18.5 and that's 19 and 19, they're going to want to include that as well. So that's fine. That isn't one that we're going to discontinue. So what they did with this is exactly like the experiment that we did. However, the one that you did for photosynthesis was changing the distance between the light and the pondweed that was bubbling away because it was going through photosynthesis in the test tube. If you remember, that's one of your required practicals for the B4 topic, and it's one that comes up often, so it is one that you need to know. So in here, we did our test tube, we had our pondweed, we had the light source, and the one that we did was using a ruler, we moved it back each time. So let's say the sunlight was 0 centimetres from the pondweed, then it was 5 centimetres from the pondweed, then it was 10 centimetres from the pondweed. That's not what they've done here. The independent variable, and I always think of it and tell my students, independent I know. This is the thing that you know you are changing before you do the experiment. So it always goes in this left-hand column. The independent variable that they were changing wasn't the distance, it was the temperature instead. The dependent don't know variable was the rate of photosynthesis, and they calculated that by volume of oxygen produced, it says there, volume of oxygen produced per hour. So that's how they felt they were able to tell how the rate of photosynthesis was affected. So this one, as we said, calculate the rate of the mean X. You would do 18.5 plus 19.3 plus 19.5 divided by 3, because that's how many there are. This kind of question, you need to know how to do the mean. That's not just for this, it's for chemistry, physics, and of course your maths paper as well. The student identified one anomalous result in table 1. So an anomaly is something that doesn't fit the pattern. When I have a look at this, I said to you, I wasn't sure if they were going to be really mean and say that that was too low. But when we have a look at these are all about similar until you get to this line at 45 degrees. So you can see here the data says 1.9, 2.2, and then 14.2. So this is far too high. So you would just circle that part. Suggest one cause for this anomalous result. So there could be lots of things that went wrong here. When we have a look at 14.2, that's probably more fitting for around 20 degrees, perhaps, yeah, I would say probably 14 is closer to those, isn't it? Not to 45. One of the things that can happen here, you could obviously say that the temperature could be wrong for whatever reason. Maybe, I don't know, the sun's shining through the window and suddenly the temperature's changed, or a cloud's come, I don't know. You could also have said, if you remember guys, when you moved your light, and this comes up quite a bit when you're analysing the test that we did, when you move the light source back by however, like whatever the distance was, you had to wait for 5 minutes to allow the plant to acclimatise, and that just means to get ready to to get used to the new conditions, and so that it allows it to change its rate of respiration according to the new environment. So one of the things that they could have done there was they moved it to 45, but they hadn't waited long enough, and that could just be a drop, if you can see there, from around 22, where let's say they've started timing it at 2 or 3 minutes waiting rather than waiting another 5 minutes. So just one possible cause of the anomalous result. I'm going to say either temp is incorrect, or didn't wait 5 mins to allow rate of photosynthesis to set to new temperature. How did the student deal with the anomalous result? This comes up all the time, and people say things, stupid things, because it's an exam, like, oh, they did it again. We have no evidence of them doing it again. It says test 1, test 2, test 3. We don't know about a test 4. When you have a look at this mean 2.1, what they must have done was just ignore that and only use these two and then divide it by 2. So it's not that they did it again. What you want to say is that they ignored result and used only trial 1 and 3. And those are the ones that suited, you know, that those two were similar, that one wasn't. Give one factor that students should have kept constant in this investigation. Well, we know the thing in this one that they were changing, our independent variable was the temperature. Dependent was the rate of photosynthesis. What they're saying there is, what are all of our control variables? And control variables are things that you have to keep the same so that you are only testing the independent variable. It could have been things like the mass of pondweed. It could have been the species of pondweed. It could have been the concentration of carbon dioxide in the water. And the way that we were able to monitor this, if you remember, we put a carbonate source in, so something like sodium carbonate was put into the, like as a powder, was put into the water so that there's carbon dioxide to allow the plant to photosynthesize. So it could have been any of those. You could also say same light intensity. So in this one, they weren't changing the distance of the lamp, so therefore the light intensity has to be kept the same. Over here, they've put the same graph in because you're meant to be able to to use from the same question rather than turning over, which is good. Why did the rate of photosynthesis decrease from 35 to 45? So we've got this set of data to 45 here. So you can see up until this point, we calculated this obviously earlier, you've got low rate photosynthesis, and then it gets a little bit higher, gets a bit higher, and then it starts going down. I mean, is what it is, fine, don't count that. That one went up from that one went up from that one, then we've got a drop, then we've got a drop, then we've got a massive drop. So we know that the optimum here, if you remember, we said when the enzyme works in its best conditions, that's called the optimum. This suggests that 30 degrees is the optimum because that had the highest rate of respiration, because there were the most oxygen bubbles were given off here. So what happened after this point that meant less and less and less respiration took place? That's where you have to know if it's about temperature, it's going to be about enzymes. So what will happen as the temperature increased above 30, so I'm going to say above 30 degrees, the enzyme active site started to denature. So denaturation is when something like conditions are either, that's our enzyme, the active site, which is this thing here, that's where the substrate, anything that's being made up or broken down, fits into. We know that there's a specific shape between the substrate and the active site, and they will come together. If you've got a high pH, so something like pH 13, if you've got a low pH, so something like pH 2, for example, or if you've got high temperature, low temperature just means there's low kinetic energy, but it doesn't cause this to denature. That will cause this active site to denature, which means it changes shape like this. So now that substrate is no longer going to be able to fit in there. Bit by bit by bit, the enzymes in the plant will denature, so they won't all go at one point. So it's not like you went from 30 degrees to 31 and suddenly no respiration took place. Some will denature, but not 100% of them. You can see even at 45 degrees, some of them are still working. So that's why you use the word started to denature, not that they had completely denatured. Okay. Then we go on to this bit, label the y-axis, use the scale, 5 marks, perfect. For this one, guys, you can see they've already given you temperature and they've given you the units. One of the main things that people forget to do in here is label the axes and put on the units. Please write whatever has been put on this part of the table. Don't put it in your own words. Some people, for example, in this would just say photosynthesis. That's not what's being measured. It's the rate of photosynthesis in centimetres per hour. We've got this already labelled at the bottom. So your independent variable always goes on the bottom. Your dependent don't know variable is the thing that's going up the side. So rate of, and I'm just going to do that, you know what I mean. And you have to put in your unit, centimetres per hour, centimetres cubed per hour. Up here, then, obviously, you plot your points. I haven't done this. This starts at 25. Oh, so they don't even want you to do the X. Haven't looked at this, but you would obviously label your axes along here, and then you'll do some line of best fit. I haven't plotted the points, so I can't remember, or I can't visualize right now what's going to happen. I know it's obviously going to go up and down. Fine. When you've got something like that, we know that 2.1 is going to be your lowest value at 45. The highest value is going to be 44. So that's what you need to take into account when you label up here. Make sure that you're consistent with your divisions. So let's say, for example, if you're calling that 5 and that 10, don't then say that this next regular set of 5 is going to be 90, right? If you've done 5, you've got to go up in 5s, and people always kind of get that confused. When you've got this shape, that we know that it increased and then decreased when the enzyme started to denature. Don't force that into being a graph that looks like that. That's not what happened. It went up and then went down, so that's got to be, when you do your line of best fit, what we call a freehand curve. You never do a dot to dot, so that's where you get your ruler and line it up like this. I know you do in maths, you don't do that in science. It's either a line between the points or a freehand curve. Okay, on to this next bit. Diffusion is an important process in animals and plants. That could be something like gases, right? That we're breathing in, breathing out, and plants are using the gases for photosynthesis. What is meant by the term diffusion? It's a 2-marker. So the first thing you are saying, it's the movement of particles. You could say substances, that's fine, but the idea here is that it's the movement. The next thing that you want to say is from an area of high concentration to low concentration, or you could have said down the concentration gradient. So from where there's a lot to where there's not so many. We then got this question here. This was layers in the leaf. This was B2. Molecules of carbon dioxide diffuse into the mesophyll cells. So we've got CO2 outside the plant. It goes through the stomata and then it goes into these cells in the spongy mesophyll layer. Which two changes will increase the rate at which carbon dioxide diffuses into the mesophyll cells? So what the question's doing, and I would draw this out just to picture it, is saying how can we cram more carbon dioxide in there? When you start having a read down here, increasing, I'm going from bottom up, increasing oxygen concentration in the air, that's not going to change the amount of carbon dioxide going in, so I'm going to cross that out. Increase the number of stomata that are open, absolutely. If you've got more spaces for carbon dioxide to go in, that means that more is going to be able to fit through. Increase the amount of carbon dioxide concentration in the air, absolutely. If you've gone from having 8 molecules of carbon dioxide outside to having 800, of course more of it's going to be able to come in. Decrease surface area, absolutely not. Increase number of chloroplasts in the cell, those two are the most appropriate. Don't start, I mean, you could try to work that out in terms of how that would affect the rate. It's going to be these two. You don't have to put the crosses in, I've just put that in there, but you need to tick 2. And again, they've put this in bold to highlight to you that there needs to be 2 answers. Often people just tick 1 and leave it. You're never going to get marked down for a guess, so you can only, you can only gain points here. Don't just leave it or just leave 1. Any multiple choice, you are expected to put something down, just anything. Okay, this style of question comes up quite a bit. Usually it's talking about the villi and microvilli that you find in the small intestines in digestion, but this is all to do with diffusion. So it's like gas, exchange surfaces, it was called, and you did it in year 9 and B1. Diffusion also happens in the human lungs. So we know here you're going to have oxygen going from the lungs into the blood, and you've got carbon dioxide going from the blood back into your lungs to breathe out. Figure 3 shows the human breathing system. They've shown you this picture because they want you to use all of this information. They didn't just give you that, they made a point of mentioning capillary. The number of people, oh, sorry, my cat moved the phone. The number of people who just ignore this part and only kind of talk about the lungs. If they've mentioned any bit of information like capillary, you have to use it. And it's like giving you like a phone a friend. They've told you that's the thing that you need to include in your answer. Explain. So that's not just state. State would be something like, there's a capillary. Explain is you have to say scientifically how that has an effect. So it says, explain how the human lungs are adapted for efficient exchanges of gases by diffusion. So you want to keep referring back to the gases that are transported. We've already said oxygen goes from the lungs into the blood, from the blood CO2 from your cells, and where it's been produced in respiration comes back into the alveoli. So the whole purpose of this is every cell in your body has to have oxygen going in so that their mitochondria can respire. Respiration releases energy, and a byproduct of that out the other side is it releases CO2. That's a waste and we want to get rid of it. With this, the answers are always the same. They don't change. First thing, they haven't told you the word alveoli. So hopefully you remember this guy down here that looks like a grape are the alveoli. So we want to say alveoli, increase surface area. So a larger surface area means, if you can imagine, if you had a space like this, you might fit 3 molecules of oxygen across, right? In that same space, if you turn that into a grape-like folded structure, like that, which is what the alveoli look like, a molecule can go across there, it could go across there, it could go across there, oxygen could go across there, go across there, go across there, go across there. So rather than 3, you've got 1, 2, 3, 4, 5, 6, 7. Increased alveoli, increased surface area with folds means more particles can exchange. I'm not even going to explain, but I would at some point. You need make a point of saying, I could say here, O2 out of alveoli into blood, CO2 from blood into alveoli. Okay, so you can bullet point in any 6 marker, and please don't waste your time writing the question out. So, for example, I used to, when I was your age, take about 4 or 5 lines to say, in the human gas exchange system, blah, 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 blah. I'll explain, don't do that, just bullet point straight away. So the first thing we've said is increased surface area, and that just means more particles are able to move. The next thing you always want to say is a good blood supply. And this is, I think, quite hard for people to get their head around. Good blood supply is always on the mark scheme, and that means lots of capillaries. And why does, I think it's IES. I'm so dyslexic, guys. No idea. Capillaries. Why does that have an effect? Essentially, if you can imagine, if you've got an area, like a space like this, and you've just got one capillary here, capillary, and that's your alveoli, and you've got 50% O2 here, you're going to get, moving from high to low, diffusion taking place across here, right? And then you're going to reach a point where there's 25% oxygen there, 25% oxygen in there. Diffusion's then going to stop because it only goes from high to low. So by having another capillary coming close by to the alveoli, this one's going to have 0% oxygen, this capillary. This one's going to have 25, so you're even going to get more diffusion happening. You'll then get to a point where that's 12.5% oxygen. This in your alveoli is also going to be 12.5% oxygen. And then if you get another capillary coming round, it's going to go from high to low, so then you're going to get to 6 and 6%. Basically, hopefully you can see here, the more capillaries you have, the more oxygen that's going to diffuse across, and obviously carbon dioxide that's going to diffuse back across as well. So good blood supply means lots of capillaries, and the way that you explain all of this is it maintains or keeps a difference in concentration gradient. So what I mean by that is there's always a higher amount in the alveoli than there are in the capillaries around it. The next thing, so we said here, increased surface area, you've got good blood supply, difference in concentration gradient. These are the two things, increased surface area, good blood supply, that come up all the time. The third thing we're going to talk about is thin. Like you can see in here, they showed you that around the alveoli, there's this tiny thin layer of cells, around the capillary, tiny thin layer of cells. And what you hopefully remember is each of these are only 1 cell thick. So alveoli wall is only 1 cell thick. And capillary wall also only 1 cell thick. So I'm going to say thin. So smaller distance for diffusion. I'm going to highlight this in a different coloured pen just so you can see difference from there. So we've said thin, smaller distance or shorter distance. Again, that's something that comes up a lot. The last thing, and it leads on to this idea of maintaining a difference in concentration gradient that you can talk about, is you are constantly breathing in and breathing out. So if you took in one big breath and we had 100% oxygen in there, obviously you get to a point where you've got some of it diffusing through into those capillaries. If you just held your breath, eventually we'd pass out because you've got no more oxygen molecules to diffuse across. The fact that we breathe in and out, constantly ventilating our lungs, means there's a continual new supply of oxygen. So breathing, and the next one is blood flowing. Both of these things, this idea of air in, air out, blood to the around the arteries and then pumped away, blood to the arteries, not arteries, I meant alveoli, sorry. Blood to the alveoli passes away, blood to the alveoli passes away. These two things means that there's always a difference in concentration gradient, always. But again, you don't have to mention all of these to have got this 6 mark. It's just confidently being able to understand some of these points. Okay, this one, root hair cell. what we've got here, it shows you water molecules as the dots, nitrate ions as the crosses. What you should remember here is, first thing, if we're looking at root hair cell, we know water, we're going to be talking about osmosis. This root hair cell has a larger surface area. Is one of our specialised cells that you did in year 9. Nitrate ions, you know, hopefully you remember, any minerals have to go into the plant against the concentration gradient, so going from a low to high. So what we've got here, if I show you these crosses, we've only got 1, 2, 3, 4, 5, 6, 7 outside, and you can see it's really highly concentrated in there. This process is active transport. So it says, name the process by which water moves into the root hair cell. We've already said osmosis. If they haven't said, say it. If osmosis comes up every year, apart from 1 year that it didn't, only one since I was a kid, it comes up all the time. If they talk about the movement of water, don't assume that they've mentioned osmosis. Just write it in your answer. Always, 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 because it's usually always a mark. Nitrate ions need a different method of transport into the root hair cell. Active transport, and that's where it goes from a low concentration to a high concentration. Explain how the nitrate ions are transported into the root hair cell. Use information from figure 4 to help you. So your explanation here, why is it active transport and not something else? You want to say that it's going from a low concentration gradient to high. And then the last one that you want to say, how is it able to do that? That's this crazy process. How is it able to push it against the concentration gradient using energy? And I'm going to go one better just to make sure the examiner knows what I'm talking about. Using energy from respiration in the cells, or you could even say that root hair cells, mitochondria. But you only need to have said low to high and energy. On to the next one. Straight away we can see here you've got that number 1 as micrometers. This number 2, they've given that cell as micrometers. Whoa, whoa, whoa. They've given you this cell in millimeters. This is something that you did around the time that we talked about microscopes, and hopefully you remember you've got centimeters, millimeters, micrometers, and that's what you always have to come back to, because that's what that's pretty much always the size of the cell that we're looking at. Nanometers, I've only ever once seen a question where they've asked you to convert to nanometers. Those are viruses, they are so small, so this is the one that you've got to get to. If you've got 1 centimetre, to get to millimetre, you times by 10. So we know that 1 centimetre is 10 millimetres. To get from millimetres to micrometers, you have to times by 1,000. If you went from micrometers the other way to nanometers, you would then divide by 1,000. Micrometers to nanometers is times by 1,000, but again, like I said, very rarely comes up. We are shown these cells, and you quite obviously can see massive nucleus. That's in an animal, liver cell. Mesophyll we know is in a plant. This is a bacterial cell, and quite visibly you don't have a nucleus in there. Give 2 similarities between the prokaryotic cell and the eukaryotic cell. Lots of people did badly on this question, and it's really sad because it was simply that they couldn't remember what a prokaryote is and a eukaryote. I always think of it, if you think of the letter P, right? Turn it into B. Yeah, looks a bit like a B. A prokaryote is a bacteria, only bacteria. E for everything. Everything else is a eukaryote. So those are plants, that's animals, it's fungi, it's a protist like malaria. Everything else is a eukaryote because they have a nucleus. Whereas, sorry, everything else is a eukaryote. I hope I said that. Everything else, everything with a nucleus is a eukaryotic organism. Bacteria don't have a nucleus, so they are prokaryotes. So 2 similarities between these two. Well, we can see that they've got grey shading in the background in all of them. So they've all got cytoplasm. Has cytoplasm. They've all got these tiny little black dots, and those are ribosomes. So both have ribosomes. They all, They all have the first layer here, or the first line. That's the cell membrane. So you could say they all have a cell membrane. I'm not going to say they have a cell wall, because although a plant cell does have a cell wall and a bacteria do, we don't. So I'm going to leave it at that. They've all got DNA. It's just in a very different form in the bacteria. Give 3 differences between the bacteria and the eukaryote. So in this one, guys, people have lost marks because they'll mention one but not the other. So let's say, for example, these guys here, these are mitochondria. So I'm going to highlight, you can see there's a mitochondria in an animal cell, there's a mitochondria in a plant cell. So people will say things like, eukaryotic cells have mitochondria, and they haven't compared it. The question has said differences, show off to the examiner. You're not just saying these have it, you have to say, and this one does not. So if they're asking you for differences, they're asking you to compare. This happens all the time. Sometimes they do it when they ask about what's the difference between arteries and veins. And people will say, arteries have a thick muscle wall. If you haven't compared to the fact that veins have a much thinner wall, then you're not going to get the mark. So you have to say thicker wall. So differences here, we're going to say number 1, eukaryotes have mitochondria. And then the last bit you have to say is bacteria, aka prokaryotes, do not. For here, you're going to say eukaryotes have nucleus, bacteria do not. And then here, if you remember, guys, these rings, are called plasmids, and they're like these circular loops of, like circular loops of DNA. They don't have chromosomes in bacteria. So we've obviously got chromosome from mum, chromosome from dad, and that's pair 1. Chromosome from 2 from dad, chromosome 2 from mum, that's chromosome 2. Those are all in humans. They don't have that. They've got one long DNA loop. So here you can say bacteria have a DNA loop, whereas eukaryotes have chromosomes, or you can say bacteria have plasmids, and plasmids are those like hula hoops of DNA, and eukaryotes do not. Okay, then we're going to go on to this one. Calculate the ratio of the size of the bacterial cell to the size of the mesophyll cell. So we were told here that the bacteria was 1 micrometer, and that's going to be the bacteria, because it says size of bacteria compared to size of mesophyll. So we've been given our first part, which was 1 ... Then we're going to talk about the mesophyll. The mesophyll, we were told, is 0.05 millimeters, and straight away, like I said, the fact that they've put 1 in micrometers, 1 in millimeters, you knew you were going to have to use that. So I said to you guys, millimeters to micrometers, you times by 1,000. So we do 3 jumps where it's 1, 2, 3. So that will turn it into 50. Does that make sense to everyone? Hopefully. 5, 0, and then you've got the decimal point. So 1.50. Name the type of cell division that produces genetically identical body cells. So hopefully everyone at the moment is talking about meiosis in year 11 lessons. Meiosis is producing 4 non-identical daughter cells. In mitosis, you've got 1 cell that could be like a skin cell. It produces 2 genetically identical daughter cells. When we're talking about bacteria dividing, there he goes, they don't divide by mitosis because they don't have chromosomes. So they can't have their chromosomes lining up down the middle and pulled to the poles like animal cells do, for example. These guys divide by a process called binary fission. This isn't in the question. I'm just explaining this as a bit of revision to you. If you're asked how bacteria divide, it's by binary fission. Okay. This shows the cell cycle, and this is one that I definitely need to go over in a bit more detail with my classes. So I've focused a lot on mitosis, and I know we've done this as a bit of a starter with in some of them, but actually the rest of the cells, like life, if you will, you need to understand a bit about that as well. This shows the cell cycle. So if you can imagine, this is you being a baby, toddler, teenager, middle-aged, elderly, done. Yeah, so when we count up these 1, 2, 3, 4, 5, there are 20 of these. It says what percentage, so I'm going to say here segments, there's times 20. What percentage of time for one cell is represented by stage 2 and stage 3? So that and that. If you've got this whole thing almost like representing 100% of the cell's life, and we know each section is 20, if each section was 10, then if there were 10 sections, then each would be worth 10. There are 20, so each section is worth 5, yeah? Because you've got half, like it's got to be half because there are more of them to get to 100. So if we count 5, you've got 5, 10, 15, 20, 25, 30, 35. So you're ticking that. This style of question has come up quite a bit in the past. Then this is the one that I think is really hard. And again, I know that I need to go through this with my classes in a bit more detail. What happens during these different stages? So we know we've discussed in my classes anyway, the fact that your DNA has to double before then the cell can be, it can turn into 2 different cells. So you have to double the DNA and then they'll line up, pull to the poles, etc. So in this first part, number 1, that's what's going to be taking place here. The fact that you can't just have, if you're going to have a brand new cell, you can't split. So you've only got half the genetic information. You need to have 46 chromosomes in the old cell and 46 chromosomes in the new. So it has to double. So number 1, DNA doubles. And the reason that you know that's in stage 1 rather than stage 2, nothing else can happen if you don't have double the amount of DNA. So that's got to be the first one. We then know that they line up down the middle and they are pulled to the poles. So I would write down here, chromosomes line up down middle and are pulled to the poles. So this is by something called spindle fibers. In this, on the mark scheme, it actually just wanted you to mention the idea that they each move to the poles. It wasn't about the lining up part, but I'm adding that in there as well. In here as well, just to give you a bit more information, it's not just that the DNA doubles, mitochondria doubles, ribosomes double, the amount of cytoplasm will double as well. For stage 3, this is almost like, it's just about to split into 2 cells. So in the very final part, if you can imagine, you're looking like this, where that's formed, it's about to break away from one another. So this is where the cytoplasm and, can you look at that? That's got to divide, hasn't it? Cytoplasm and cell membrane divides. And you had to put both. It was really specific in this question. You couldn't just say the cell membrane divides. You had to say the cytoplasm and the cell membrane divides. And this is this final point that people always forget to write in when they're talking about mitosis. They talk about the idea of the chromosomes or the DNA doubling, lining up down the middle, pulled to the poles, and then they forget this very final bit. So cytoplasm and cell membrane dividing. Okay, 4.1, lipases break down lipids. So just to remind you, anything with A's on the end means enzyme. So a lipase breaks down lipids, a protease breaks down proteins, a carbohydrate breaks down carbohydrates. Sorry, my rabbit was just eating our rug, so I just had to go push him off. Right, here we go. So what we've got here, I've just drawn out a lipid molecule. This part is a glycerol. Glycerol. Can't spell it, but you don't need to in biology. Spelling, is not English. It's okay if you spell things wrong. These are 3 fatty acids. And for those of you who are doing separate sciences, you need to remember a practical that you did where you used an indicator called phenolphthalein to indicate when the solution had become acidic. And that showed you that fat was breaking down because the fatty acids detached from the glycerol molecule and the pH of your solution lowered. For those of you who are doing combined science, you didn't need to know that one. So when I have a look at this, it says which 2 products are formed when lipids are broken down. We've said, glycerol and fatty acids. Amino acids are found in a chain of proteins. Glucose, we know that that's a sugar, so that's got nothing to do with fats. And glycogen, we've now done in paper 2 when we did homeostasis and controlling blood glucose levels. And you hopefully remember that there's a store of glucose in your liver as a molecule of glycogen. So those are the two. Again, don't just tick one. If you're in an exam and it says 2 and it's made a point of putting that in bold, make sure that you tick 2, even if you're not sure, you don't remember, at least you can get 1 mark. For this one, enzymes comes up every single year. Please make sure that you know it. It says, one molecule used to explain enzymes is the lock and key theory. Figure 7 shows a model of the theory. So what's important to me here, I suppose, they haven't just said, describe the lock and key theory, which they've done in exams before. What they've done is included this picture. So you're going to need to use this diagram or this picture when you explain your answer over here. It says, explain the lock and key theory of enzyme action. Use the information above in your answer. They've, as I said, made a point of showing you each step. You are talking about, and I don't think this was a mark, but I'm going to add it in, that the enzyme is like a lock. The substrate is like a key. This has come up before where you've had to put this mark in and explain in this theory which part is which, but it's the next part that was on the mark scheme. Substrate has a specific shape that matches enzyme active site. So again, that active site is where the magic happens. If I've got a key to my house, I cannot enter the school because the lock is a different shape. And it's the same with these enzymes and substrates. Only certain keys will fit certain locks. Only certain substrates will fit certain enzymes. So you have to always talk about the specific shape. And again, we've said in the active site. So you can see there, they've made it very obvious that it's a really weird shape and that weird shape fits that weird shape. If we're then using this information from the diagram, that's fine, we knew that already. I want to have a look at what's being said here. They've made a point of saying substrate products, okay? So we're going to talk about this bit here. What has happened between substrate and product and why they made a point of this adding this in. So out of 3 marks, your first mark is by talking about those. The next mark then is going to be talking about like what's essentially the changes that are or aren't occurring. So the second thing I'm going to say is substrate is broken down, and we can see that here, into product. And the mark on the mark scheme is about the idea that it's broken down. It's no longer as it was before the experiment started. 3, what about the enzyme though? Enzyme is, and you could have said unchanged, like the shape doesn't change. One of the things that keeps coming up in unchanged in shape, one of the things that keeps coming up, and it was on the allow section in the mark scheme, you could have said not used up. And because this comes up more like what is an enzyme, or can you explain a biological catalyst, and you have to say the enzyme is not used up in the reaction, I'm going to highlight this as a point, the fact that yes, it is, right, it's still there, it hasn't been damaged, it's not used up, you can keep using it again and again, but as you can see, the shape also hasn't changed. There are many different types of lipases, so lipid enzymes in the human body. Why does each different type of lipase act on only one specific type of lipid? So there are lots of different types of lipid in the body, or in the foods that we eat. If you can imagine, you've got very different oils in avocados than you do in fish. Again, why are there different lipases acting on different substrates is all to do with the shape. So different lipids have different shapes and will not fit into all active sites. So again, that idea of specificity, can't say it, how specific it is between one shape of the substrate and the shape of the enzyme. Okay, students investigated the presence of both starch and glucose in the leaves of geranium plants. This is the method used. So I'm just going to remind you that glucose is a 6-carbon molecule made in photosynthesis. It can be stored as starch, starch for storage in the leaves. And there are 2 different food tests that you can use to test for these, which we'll come on to. This is the main method used. They use 2 identical. So again, this will be a control variable. The idea that they are identical, so you would hope that they are the same species of geranium, the same age, probably cuttings taken from the same plant. They should be the same mass, on a bench near a sunny window for 2 days. So you've got sun for 2 days. After 2 days, leave one plant near the window. So I've got sunny window for 2 more days. And then another one, place one plant in the cupboard with no light for 2 more days. So we've allowed it to photosynthesize for 2 days. Then you've allowed one of them to photosynthesize for 2 more days. So it's had 4 days in the light. One of them's just gone into darkness. Remove 1 leaf from each plant, crush each leaf to extract liquid from the cells, test the liquid for glucose and for starch. Describe how students would find out if the liquid from the leaf contained glucose. So again, this is reminding you of your food test that you did. Testing for glucose, the first mark you're going to get is always a 3 marker. Mark number 1 is for saying you use Benedict's solution. So that's the first mark. Second mark, you are saying it goes from blue. Please make sure that you say what it goes from and to. They've been really particular about that before, where you couldn't just say goes orange. And if you remember, this is the one where it starts blue, and if there's only a little bit, it goes green. If there's a little bit more glucose, it goes orange. And if there's tons, it goes brick red. For this one, it's a plant. I'm going to say it's probably around orange, but you could have put any of these in here. So it's from blue to orange. The third one, people always forget this mark. You have to say, do reaction test in a hot water bath, or you could have said boil it. /boil. It's anything that basically is like 65 degrees or over. It's not going to work if you don't do that. So if you just drop some Benedict's in a solution in, like, you know, I don't know, on a Mars bar, it's not going to change colour at all, because it has to be in really, really hot solution for it to happen. So your 3 separate marks: the solution, the colour change from and to, and the fact that it has to be in like hot temperature. Describe how the students would find out if the liquid from the leaf contained starch. Starch and glucose are carbohydrates, but they are different types of carbohydrates. We would call one a simple sugar and one a complex carb. So our starch, if you remember, is the iodine test. And this one, you don't need to use a hot water bath. So you're just saying that it goes from orange to blue-black. I don't write the blue part because for me there are 2, like Biuret to test for proteins is blue, Benedict's for test for proteins is blue. If you just wrote blue here, you wouldn't get the mark. So although some people call it blue black, just write black. If you just write blue, you won't get the mark. Just black, you will. So orange to black. Then over here it says table 2 shows the students' results. The one that was left in the light for 4 days, so this one, if you remember, had sunlight for 2 days and then the window for 2 days, had lots of glucose and it had lots of starch. Or it had starch. The one that had 2 days in the light and then 2 days in darkness had basically no glucose and absolutely no starch at all. Explain why the leaf in the light for 4 days contained both glucose and starch. 2 marks, you're going to have 1 for each for explaining. So your first mark, why has it got glucose? It was able to photosynthesize in the sunlight. Okay, sunlight was there, it was able to photosynthesize. Something that's made obviously by the plant other than oxygen is glucose in photosynthesis, so it was able to do that. Number 2, why did it also have starch? Some glucose was stored as starch. It's like me on Christmas Day, right? I'm going to be eating tons of sugar, so I'm going to have sugar in my bloodstream. But I'm also going to, some of that sugar, I'm not going to be using it right away on the day. I'm probably going to do very little walking around. It's going to be stored in my body as glycogen and then converted into fat, ready for the new year. Explain why the leaf left in the cupboard with no light for 2 days did contain glucose, and we can see though that it's just some, only a tiny bit, but did not contain starch. So it's a bit like saying, if I've then had Christmas, I've eaten tons, and then I've gone on a calorie deficit for 4 weeks afterwards, why have I no longer got fat stored in my body? What would have happened, we were saying, there is no starch. If you look over here, negative, no starch. No starch because it was broken down to glucose. So why is there only a tiny bit of glucose, not tons? If we've said all of that like starch was broken down that the plant had, why is there only a little bit? You're saying glucose is used by plant for respiration or active transport in the roots. Or you could have said, or for growth. Yeah, so that's why you didn't have like 100% glucose. It was just kind of like breaking it down, whatever. And then the last point for 3, in dark, the plant cannot photosynthesize. So it's relying on any of the glucose that it stored as starch earlier on when it had the 2 days in the light. Suggest one way the student could develop the investigation to find out more about glucose and starch production in plants. Weird question. So what we're saying here, what could they essentially do differently? Well, the first thing, weak positive, weak negative, that's not very like specific. They could do a test to get a numerical value. So you could, like, that's one of them. You could say, be more accurate. By getting like a colorimeter or whatever and getting a numerical value. The next thing you could write in here that they could investigate using different species of plants. They just said geranium species, I can't multitask, of plant. You could have used a different one. You could have said that they could have been left in there for longer. So we obviously only had 2 days in, 2 days out. You could have seen what the result would have been after a week. Okay, many plants have evolved defense mechanisms. Figure 8 shows part of the gorse plant and part of a deadly nightshade plant. The gorse plant has evolved to have sharp thorns. What type of defense are sharp thorns? They are physical. I think this is only separate. If you're doing combined, I don't believe you need to know physical and chemical. So for this one, you could have said physical or mechanical. How do thorns defend the gorse plant? Defend the gorse plant. You can't just say that they hurt animals because, brushing past them, they might hurt. It's the idea that an animal will try to eat them once, they'll hurt their mouth and they're never going to do it again. So herbivores try to eat it, get harmed, and won't try to eat again in future. The deadly nightshade plant has poisonous berries. What type of defense response is poisonous berries? It's chemical. So if you're talking about digestion, a physical breakdown of food is something like your teeth mashing it up. Chemical digestion are the enzymes that will break it down on a molecular level. A scientist noticed that one area of the gorse plant has yellow leaves and stunted growth. So this is definitely for separate hire. So if you're doing combined, the rest of this paper is not for you. Yellow leaves and stunted growth. One reason for yellow leaves and stunted growth is a deficient of nitrate ions. Explain 2 other possible reasons of yellow leaves and stunted growth. So this is in the immune system topic where you talked about deficiencies in plants. The ones that you had to remember, I always remember Sunshine, right? That makes me think yellow. So a leaf can go yellow if it doesn't have magnesium, right? Magnesium is an iron. I always think magnesium sounds a bit like magnum. Magnum you have in the sun is yellow. What I'm trying to say here is if there's a magnesium deficiency, so it doesn't have it, the leaf goes yellow. And that's the only way in my head it makes sense. Magnesium sounds like magnum, you eat it in the sun is yellow. If there's no magnesium, the plant goes yellow. So reason one, magnesium deficiency. Oh, I spelled it wrong, whoopsie. Explanation, cannot make chlorophyll. So chlorophyll. So chlorophyll is obviously that pigment in that molecule in chloroplast that will absorb sunlight. To make it, you have to use magnesium. If there's no magnesium, it won't happen. What's another reason? I'm going to just refer back to this idea of nitrate, just to remind you that nitrates are used to make amino acids. So if it can't make amino acids, it can't make proteins. And if it doesn't make proteins, it's going to be really small. If a human being couldn't eat protein, we'd be tiny. So again, that's why something might be smaller and yellow if it didn't have like nitrates, it couldn't make the proteins. So another reason, this is referring back to when we talked about rose black spot and the different types of diseases. So if you remember, you had a plant, and if it has this fungal infection called rose black spot, it has black spots of fungus and then the leaf goes yellow. So I'm going to write here, it could have rose black spot fungal infection. And this, in this case, the black spots block the sunlight. Fungus stops plant getting sunlight, so turns yellow as no photosynthesis. You could also here have said you could also here have talked about aphids. So hopefully you remember aphids are these little nasty bugs, right, that are often found on things like roses, and they pierce their mouthpiece into the phloem where glucose is transporting up and down for the plant. When they take the glucose from the plant, it's not able to go through all of the processes it needs to, so it can go yellow and it will die. In here, really simply, and I wouldn't have thought to have said this, but it was on the mark scheme, you could have just said that maybe they weren't in sunlight. So one of the reasons it might go yellow, if it's not in sunlight, it's a bit like the leaves in autumn. The chlorophyll breaks down and it's going to die or drop off because there's not enough light. This is a really hard one. It's something that is true. I had a plant that had this. It's called the string of hearts, and they're really quite, they've got like heart-shaped leaves, so they're really pretty, but they've also got these things called nodules. Nodules you're not meant to have known about. It's just giving you a hypothetical situation that you have to talk about. So what happens in here, you've got a plant, the plant grows these things called nodules, and the bacteria can live in there. Bacteria are able to convert nitrogen gas from in the air. So we know that that's the highest amount of gas in the air. It's like, what, 79% nitrogen in our atmosphere, something like that. And they can convert it into nitrogen ions that a plant can use. So humans and plants, we can't use the gas nitrogen. We have to have it in like a, like solid form. So the bacteria can work with the plant by converting it from gas into like a solid, nitrogen ion. Suggest how the nodules benefit the bacteria. So what are the bacteria getting from the plant in these nodules that's going to be beneficial to them? Bacteria, we know, need a food source. So you're going to say that, and again, this is so tricky, bacteria use plant glucose. So this is the benefit to the bacteria. They're able to use the glucose that the plant has made, plant glucose from photosynthesis. Why do bacteria need glucose? And that's your second mark for saying they use it for growth, going through binary fission, they use it as a metabolic substrate. Explain how, and that's your growth or whatever, what it's used for and what they're there for. Those are your 2 marks. Explain how the nodules benefit the gorse plant, or the gorse plant. Attracts bacteria, because that's where they live. Bacteria make nitrogen ions available to plant. And now you need to know, why do they need nitrate ions? And there are a couple of things that nitrate ions are used for. The first one, it makes amino acids, which makes proteins. And the next thing you can say is that it's used to make DNA. So if these bacteria are dividing, they have that DNA loop in the plasmids. Nitrate is needed to make the bases, which is something that you're talking about in paper 2 now. Okay, 5.1. This is one that everyone needs to hear. So if anyone from Combined is still listening, this is one that you need to know. For many years, drugs have been distracted from plants. Which plant material was chewed as a painkiller? It is willow bark, that's an aspirin. Berries, nothing. Foxglove leaves is a drug, or makes a drug called digitalis, which is a heart medicine. Rose petals, again, do nothing.